excited today to talk about this project. I've titled this talk, Outer Hair Cell Ribbons Increase in Number and Size After Noise Damage. We're going to go through all of those terms, so hopefully you kind of understand what I'm talking about by the end and why I'm excited about it. I'd like to thank Brian for the invitation to give this talk. I've measured this out to be about 30 minutes, so we should have time for some questions afterward. And yeah, so I'll just go ahead and get started. I first wanted to start with acknowledgements. I specifically want to acknowledge my co-authors on this project, Nate Nowak, and I'm highlighting his PhD here because he just graduated in our lab. And then Kira Mull, who is an undergraduate who was really talented and worked on this project. A former postdoc of the lab, Adam Goldring, Dr. Mohammed Lahar, who took some of the beautiful electron micrographs that you'll see in this presentation, and Paul Fuchs, my advisor. I'd also like to thank other members of the Fuchs Lab and the Golatsky Lab because they've really given input throughout all of the projects that I work on. I'd like to thank the funding sources, the Center for Hearing Imbalance Training Grant and the NIDCD. And this work was recently published, actually earlier this month, and it's open access. So at the end of this presentation, if you want to kind of really dive into the details of everything that I tell you about, you can go find that online. So I really want to start with talking about how sound waves are converted to action potentials. Action potentials are just the impulses carried by neurons that convert the information from sound waves to something our brain can understand. And so we look here on the left and see kind of a basic illustration of what the outer, middle, and inner ear look like. And we know that sound waves enter the auditory canal and they hit the eardrum, which is what Dr. Narayana just gave us a wonderful talk about. And then these vibrations of the eardrum travel through the 3 bones of the middle ear, and those vibrations are converted into vibrations on the oval window. And then those vibrations are carried throughout the cochlea. So the cochlea is this spiral structure that is in the inner ear. And then the information that I just mentioned that comes from those vibrations is transmitted down the 8th nerve to the brain. The cochlea is in this spiral structure. And so if you were to kind of take the top of the bone off of this spiral structure, you would see something like this arrangement. In the blue are going to be the actual cells that detect sound. And in gray and black are the neurons of the inner ear. And there are 2 types of neurons in the inner ear, and we'll get to that in a moment. The first thing I want to draw your attention to in this kind of spiral structure is that there's the apex, which is the very tip of the structure. It would be here in this illustration, and the base. And along the spiral, there are, at different places along the spiral, you hear different tones. So if you're at all musically inclined and if you can think about a piano maybe, the low tones on a piano that would be played by the left hand would kind of be analogous to this area of the apex of the cochlea, and the high tones that would be played by the right hand would be sensed by the base of the cochlea. So now let's look closer at what all of these cell types actually look like and what their function is in hearing. So if I want to draw your attention to this dotted line, if we were to take a cross section through this spiral, then we see something that looks like this bottom picture here to the right. So there are 4 cells here in blue, and these are the hair cells of the cochlea. There's one row of inner hair cells around the entire spiral, and there are 3 rows of outer hair cells. Below the inner hair cells are these gray neurons here, and these are type 1 neurons, and they only connect to the inner hair cells. And the last thing that is illustrated in this diagram is this black neuron, which connects to the base of the outer hair cells. So it's important to remember that if you lose inner hair cells or you lose these gray type 1 neurons, you lose the ability to hear. The inner hair cells are the ones that actually detect the motion of the inner ear and send that information to the type 1 neurons, and then that sends that information to the brain. The outer hair cells amplify sound. You can think of this kind of maybe with age-related hearing loss. If you lose outer hair cells, you can still hear, but you need a higher loudness of sound to hear the same sound that you would have as a younger person. And the type 2 neurons here at the base of the outer hair cells, we're still really trying to understand their function. I'll go into it a little bit more in depth later, but there are still many open questions about how the outer hair cell and the type 2 neurons interact. And so this is really the focus that this talk is on today, is trying to understand the interaction between outer hair cells and type 2 afferents. Just because you're going to see multiple views in this presentation, I want to also draw your attention here, this is just an inset from this diagram. So if you were to look top down on the epithelium, you will see 3 rows of outer hair cells and 1 row of inner hair cells. And again, this is the side view where you see 3 rows of outer hair cells and 1 row of inner hair cells kind of opposed to each other. Once again, just to reiterate, the type 1 neurons only connect to the inner hair cells, and the type 2 neurons are much smaller in number, but they connect to the outer hair cells. So since I mentioned that inner hair cells are really important for actually hearing, let's look first at inner hair cells and talk about how they allow us to hear. So at first, if we look at this cartoon of an inner hair cell in green, you see at the very top of the inner hair cell, there's a row of stereocilia that look like small hairs, and that's one of the reasons why hair cells have the name hair cell. And what happens is when there's a vibration in the inner ear, these hairs get moved to the right, or they just get moved. And that motion gets translated to this structure here at the bottom of the inner hair cell called the synaptic ribbon. So the synaptic ribbon is this oval structure here, and these little circles that are surrounding this oval are synaptic vesicles. So when there's motion of the stereocilia here, one of these synaptic vesicles will be released from the synaptic ribbon, and the contents of that synaptic vesicle will cause a type 1 neuron here in red to fire an action potential. And so this is how hearing happens. You convert motion to a synaptic vesicle that activates a type 1 neuron. And this cartoon is beautiful and shows this perfect little round structure. But one thing we're going to look at further on in this presentation is a very up close view of this ribbon. So let's look at one and see what it actually looks like, but very zoomed in at 60,000 times magnification. So that's going to be in these 2 images here. They're the same image, but in the top, I just want to orient you by having the same colors here. So everything that's in light green in this corner is the inner hair cell, and everything that's in red here is the type 1 afferent. So this is an electron micrograph. Like I said, it's very zoomed in. And what you're seeing here, this structure that's very dense and dark, is the synaptic ribbon. And you see that this inner hair cell synaptic ribbon is surrounded by very tiny circles, and each one of those circles is a synaptic vesicle. And this is what we expect a inner hair cell ribbon to look like at this level of magnification. So this is one of the ways that we kind of check on ribbons and see what they look like. So now I'm going to zoom you out a little bit, back a little bit to look at an entire inner hair cell. So now we're looking at several inner hair cells at once. And the other way that we can look at ribbons is by staining them for a protein that forms part of the ribbon called CTBP2. And that ends up just showing up as red dots at the bottom of the inner hair cell. So hopefully you can see here each of these inner hair cells is gray in color. The nucleus is here in this large red circle. And then each one of these individual little dots at the bottom of the inner hair cell would be a synaptic ribbon. And much like this cartoon shows, every single one of these synaptic ribbons would have its own type 1 afferent that would be on the other side of the cell membrane. And so that's illustrated here in this image. Now the inner hair cells are marked in green, and this is just a row of inner hair cells. And you can see these red fibers at the bottom of the inner hair cell. And each one of these fibers would have a ribbon that would be inside the inner hair cell in a 1-to-1 ratio. Okay, since we know that inner hair cells are responsible for allowing us to hear, then what do we know about inner hair cell ribbons after noise damage? If you play loud noise at varying intensities, but anything really over 100 decibels of sound, you can actually lose ribbons at the bottom of the inner hair cell, and you can also lose type 1 afferents. And the way people have quantified this in the past is by looking at inner hair cells and the same stain that I showed you previously, where we could see the red tiny dots of ribbons. Here I've illustrated what would be the outline of maybe one of these inner hair cells. And you can see that this is what a typical staining looks like. This is a control animal, meaning that it was not noise exposed. I'll use that designation throughout the talk. And you can see that there are maybe somewhere between 10 to 25 ribbons here in this inner hair cell, and that's about the number we would expect. After noise exposure, I hope that you can appreciate that in this animal, there are now less of these synaptic ribbons in this inner hair cell. So this is a well-documented thing that can happen with noise damage. And this goes hand in hand with the fact that after noise damage, you can also have increased hearing thresholds, meaning that you also lose your hearing. So what do we know about outer hair cells and their synapses with type 2 afferents? First of all, just to orient you, we know, once again, that in the inner hair cell, each ribbon has its own afferent that it communicates with. The outer hair cells are a little different. The type 2 afferents contact many outer hair cells, and not every one of their contacts is next to a ribbon in an outer hair cell. Furthermore, when we look really closely, this is again at 60,000 times magnification, at an outer hair cell ribbon, we can see that there's the same dense body of the outer hair cell ribbon, and we can see that there's a cloud of vesicles, but it's not this tight correlation of synaptic vesicles anymore, this tight halo like we saw in the inner hair cell. Instead, it's a more disorganized cloud of vesicles. And this has been made into a cartoon just so you can kind of see it maybe a little bit more 3D here, where this would be the bottom of the outer hair cell, and this would be the ribbon, and this would be its kind of cloud of vesicles, so it would be around it. This would be the type 2 afferent. And the outer hair cells are also interesting because there is only really space for type 2 synapses and synaptic ribbons below the nucleus of the outer hair cell. So I had shown you some images of how we can look at multiple inner hair cells at one time and see all of their ribbons, and how we can then count them. And this is an example image again here. It's a little bit faint, but this is about 4 or 5 inner hair cells, and these dots at the bottom of the inner hair cell are all their synaptic ribbons. If you were to do the same stain to look at ribbons, where you should see these small red dots that represent ribbons, then you would see that each outer hair cell has a few dots at the bottom of its at the bottom of the cell. So here we're looking at the 3 rows of outer hair cells. Hope that you can see this pretty clearly. And each of these kind of vague red dots has like 2 or 3 small bright red dots in the middle, and those would be the outer hair cell ribbons. Hold on one second, I'm having a power issue. Okay. Okay, well, we will try to keep going on. My computer may die, and if so, I think Brian will have to take over with my presentation, and I will try to do it from an alternate computer.
Okay.
Let me try one more thing. Oh, there we go. That's the problem. I think. Yes, there we go. Okay, I apologize so much. So let's continue on. So what we stopped is that outer hair cell ribbons look a little bit different than inner hair cell ribbons, and there are only a few per outer hair cell, and they are located at the base of the outer hair cell. So now what do we know about their synaptic partners, type 2 afferent neurons? We know a few things about how type 2s are shaped differently than type 1 afferents. This is a good illustration of a type 2 afferent that's been filled, where you can see it has a dendrite that projects out to the outer hair cell region of the organ of Corti in the cochlea, and then it extends along the spiral for many, many microns. And what's interesting about this is you'll see this inset in B, there's a portion of the type 2 afferent that extends that doesn't actually connect to any outer hair cells. But then near the end here, you can see these tiny branches that come off of the type 2 fiber, and each one of those tiny branches will connect to an outer hair cell. And so this is what I meant earlier by saying that a single type 2 afferent can contact up to 24 outer hair cells. As I mentioned previously, there are some of these contacts, even though they're attached to an outer hair cell, they don't have a ribbon. And we would call that a ribbonless synapse. So here are a few other things that we do know about type 2s, even though we're still learning more about them. We do know that like type 1s, type 2s can respond to the contents of synaptic vesicles. But we know that type 2 neurons don't seem to respond to sound. So then the question is, if they don't respond to sound, then what do? Why are they in our inner ear? Previous people in the lab have found that type 2 afferents respond to outer hair cell death and tissue damage, which is very interesting. And then more recently, we've seen that type 2 neurons express genes that are in common with pain neurons in the skin. And so if it could be possible that type 2s report tissue damage from the inner ear, which is also the function of most of your pain neurons. One of the reasons that you can experience pain is because those neurons are telling you that there is a problem. And that would be good in a normal function because it would tell you that a sound could be damaging to your ear and you should avoid it. So this comes back to the main question for this project. How does noise damage affect outer hair cell ribbon synapses? So this is kind of the overall plan for how we perform these experiments. At day 0, we exposed mice to 100 decibel sound for 2 hours. That's the level of sound of a car horn or a live concert. And then we waited 7 days and we tested their hearing and we counted the ribbons in their inner hair cells and outer hair cells. I mentioned this before briefly, but the cochlear spiral is arranged such that at different locations along the cochlear spiral, it's responsible for hearing different frequencies. And so specifically, we took images of the cochlear epithelium at 8 kilohertz, at 16 kilohertz, and at 32 kilohertz. So we looked at very specific areas of the cochlea to kind of make sure that we were sampling various parts of the cochlea. And our noise was kind of a white noise that, that should have affected the cochlea from here to roughly here. And when we do noise damage, it's important to, especially at this sound level, it's important to note that the sound energy should damage the cochlea all the way through to the base, because the base is very preferential to be damaged by noise. Over here, I'm not going to go into this too much, but I just wanted to tell you that at our 7-day time point, when we tested the animal's hearing, we saw that they needed 40 more decibels of sound to hear the same noise. So this basically means that that the control animals have hearing thresholds where they can hear either a tone or a click at 20 decibels, which would be around the sound of leaves rustling. And after we do noise damage like this, it was 2 hours, 7 days later, they would need to hear that same level, that same type of stimulus at the sound of a normal conversation. So they would need a lot more loudness to hear the same sound stimulus. So we did induce hearing loss in these animals. So then going forward, I had talked a little bit about how we needed to be able to count the synaptic ribbons at the base of the outer hair cells, because we think those are the ones that are functional for contacting the type 2 afferents. So I'm going to discuss a little bit about how we did our method of counting ribbons. So here you're going to see, as I've shown you previously, you'll see a row of inner hair cells here along the bottom, and this is going to be a video. And then here along the top, you'll see 3 rows of outer hair cells. We did this counting in virtual reality, and then we were able to do this quantification and generate this movie. So you'll be able to see both the top-down image of the tissue and also a side view of the tissue. And what I really want to highlight here is that again, hair cells are all in green. The ribbons were stained with the same thing that I've shown you previously, where all the ribbons should show up as tiny red dots. And then we have put on these cartoon dots to count those ribbons. And for the inner hair cells, those dots will be yellow. And for the outer hair cells, the synaptic dots will be cyan. So here you can see the inner hair cell ribbons at the bottom. And as we move to a side view, you can see that these cyan dots are at the bottom of the outer hair cells. And this method just really gave us an ability to, when we were doing the counting, make sure that we were counting exactly the small red ribbons in the place that we thought that we were counting them. So I mentioned before that people had looked at inner hair cell ribbons after noise exposure. And so the first thing that we did in our data was to look at the inner hair cells and count the ribbons in the inner hair cells to see if it agreed with what had already been published. So here is a section of inner hair cells from a control animal, meaning it was not exposed to noise. And I hope you can appreciate in these white inner hair cells that at the bottom you can see the red dots that indicate the synaptic ribbons. And below is an example of an image of inner hair cells that are in white again, and they're red synaptic ribbons. And it is a little bit difficult to appreciate, but they are reduced in number. And so when we count all of the ribbons at different frequency regions in the cochlea, we see indeed that after noise exposure, there's a reduction in ribbon synapses of the inner hair cells. So this just confirmed to us that our noise exposure was working, and we had already shown that it damaged the hearing of the animals, and now we're showing that it damaged the number of ribbons at the inner hair cell synapse. But what about the outer hair cells? That's what we're really interested in trying to understand. So first, this is looking top down at the 3 rows of outer hair cells. And this is the control here along the top. And this is an example image from a noise exposed animal here along the bottom. And so I'm showing this to you in 2 ways. The red dots, again, are the synaptic ribbons. And when you look top down, you can appreciate that there are red dots here in the control, and there are also red dots here in the noise exposed. But when you look at a side view, you can see that in the control animal, for these 3 outer hair cells, there are 2 ribbons for 3 outer hair cells. But when you look at the same below the nucleus of 3 noise exposed outer hair cells, you can actually see what look like 4 outer hair cell ribbons. And so when we went and counted from multiple animals and from 2 different experiments, we saw that there was an increase in the number of ribbons at the base of outer hair cells. And this is really shocking because this is completely in contrast to what happens in the inner hair cells. Furthermore, when we counted over 500 outer hair cells for the control animals and the noise-exposed animals, we saw that on average, the noise-exposed animals had outer hair cells that had 2 or 3 ribbons per outer hair cell. And so this was also interesting because it just meant that that more outer hair cells had ribbons, and more of those outer hair cells had 2 or 3 ribbons. So then we went and looked using electron microscopy very closely at these ribbons in the control and noise damaged tissue to see if these ribbons looked any different. And so again, this is very high magnification. The top row is going to be a control animal. The bottom is going to be a noise exposed animal. In this first image, just to orient you, this is the bottom of the outer hair cell. And this in red here is the type 2 afferent contact. And here is a synaptic ribbon. And as I mentioned previously, there's kind of this dark center, and then there's a cloud of vesicles that are around, synaptic vesicles that are around this ribbon. And over here on the right, since this was done by sectioning through the tissue, we're able to reconstruct the 3D volume of what this ribbon looks like. And it looks like this, where there's a central ribbon structure, and then there's a cloud of vesicles surrounding it. We were surprised to find in the noise exposed that we had a larger variation of how the ribbons appeared. And so again, in this micrograph, everything that's in green above this line is the outer hair cell. And here in red is the type 2 afferent contact. And here is a ribbon that is very large, but it has that characteristic dark electron dense body. And it actually even has some kind of windows in the middle of it, which is definitely different. And then surrounding the ribbon, there are still synaptic vesicles. And so then that's represented here in this, this cartoon of the whole ribbon. So what was really interesting from our electron microscopy data is that we also saw an increase in ribbons in this one mouse that we were able to look at. And not only did we see an increase in ribbons, the ribbons were overall increased in size as well. And we're not sure exactly what the increase in size would mean, but it was definitely an interesting observation. So finally, I told you previously that the type 2 afferents seem like they could be similar to pain neurons. So we didn't directly test how type 2s changed after noise damage, but we still wanted to try to model the effect of an increase of outer hair cell ribbons on type 2s. So in other words, what would happen if you increase the number of outer hair cell ribbons? Would that affect type 2s? So the first thing we did is we looked back in the literature, and we had known from this one paper by Martinez Monedero that there's an average of 24 outer hair cells per type 2, and I had mentioned that previously. Next, we also looked back at some data from a former graduate student, Kat Weiss, in the lab. And what she did is she went and stimulated the top of the stereocilia, the hairs on top of the outer hair cells. She went and stimulated a lot of outer hair cells while recording from a type 2 to try to understand how many synaptic ribbons were associated with the typical type 2. And so here on the right, you can see this, this string of, triplet circles. And this represents outer hair cells. And so each one of these that are colored in a color, she stimulated. And each one of that's red, she determined had a ribbon that was associated with the type 2 she was recording from. And so overall, she came to an average of 9 ribbons for type 2 afferent. And I mentioned earlier that not every type 2 contact has a ribbon. And so this makes sense. There might be 24 outer hair cells per type 2, but only about 9 ribbons. So then Nate Nowak, who was one of the co-authors on this work, went through and tried to mathematically model this. So in his model, a control using our numbers of counts for outer hair cell ribbons, a control animal would have about 10 ribbons per type 2. And after noise exposure, based on our data, a noise-exposed type 2 would be associated with 12 ribbons. So this is an increase of about 20%. So that might make sense, right? There are now more outer hair cell ribbons, so there are more ribbons per type 2. But our next question was, could an increase of ribbon in the number of ribbons affect type 2 firing of action potentials? And so to do that, we also took some previous things that we knew from the literature. Number one, we know that each ribbon has a 25% chance of releasing a vesicle. And type 2s are a little bit different than type 1 afferents. They need 7 synaptic vesicles at one time to fire. So if you think about this, if you assume that all outer hair cells were maximally stimulated at one time in a control animal, as seen here in the black bars, we could assume that the population of type 2s would fire 22 action potentials. But if we increase the number of outer hair cell ribbons after noise damage, for instance, we see that this would increase to 44 action potentials. So in other words, after noise damage, if you were to maximally stimulate all the outer hair cells, then this would double the number of action potentials coming from the pool of type 2s in the inner ear. And this is really interesting because the whole idea is that, or one of our hypotheses is that type 2s could be reporting damage, which could be perceived as painful. And so if you are now increasing the number of action potentials coming from type 2s, then you could be increasing the perception of pain from the inner ear. So this is, like I said, this is a really interesting project, but there are definitely a few caveats, and there are a lot of still open questions that we're really interested in. I'll go through these briefly and discuss them, and then we can discuss them more in a question and answer session after this if we need to, or if you'd like to. So first of all, I mentioned previously that type 2 contacts on outer hair cells were sometimes ribbonless. Is it possible that after noise damage, for some reason, those ribbonless synapses now have gained ribbons? Is that one of the mechanisms for how this is happening? And we don't know that. That would be very interesting to look at. We also looked at one time point, 7 days after noise damage. So one question is, does this effect persist? Do the ribbons, this increase in ribbons, does that stay there, and how long? And another kind of question that goes along with this is when does the increase in ribbons happen? Does it happen at 7 days, or does it happen earlier, sometime between the actual noise damage event and when we are seeing this at 7 days? Could we have caught the ribbons in the middle of merging or splitting? We know that in development, the ribbons, both in the inner hair cell and outer hair cells, can merge and split. So is it possible that something like that happens? And also, very interesting, what sound level causes this increase in outer hair cell ribbons? If we were to increase the noise even more to the point where we permanently deafen the animals, would we still see this? Or if we were to reduce the sound level for our noise damage, is there a point at which we wouldn't see this type of effect? And lastly, just to kind of bring this up, because we talked about it a little bit in the previous talk as well, this is not the only thing that happens after noise damage. There's inflammation of the inner ear. There can be cell death if the noise exposure is loud enough. The outer hair cells can actually die. And it's possible that type 2s could become hypersensitive of their own accord from being exposed to inflammation or cell death or anything else. So how do all of those processes affect outer hair cell ribbons and type 2 neuron firing? And how do all of these processes also affect something like hyperacusis? That's kind of all an open question that we're still trying to understand. So with that, I will, I think, stop sharing my screen if I can figure out how. And then I can take some questions. Brian, I'm not sure if you're saying anything, but I cannot hear you.
Okay. Thank you. Yes. First of all, it's amazing work. Thank you so much for all of the work your lab has done. Really breakthrough, really novel. And your list of questions align with the list of questions submitted. So I think I know the answer, but we'll read a couple of them. How long after noise exposure were the ribbons observed? And is this from inflammation? Do they ever go away?
Right. So as I mentioned, we looked at one time point. So we're not sure if they ever go back down in size or number. And we're also not sure if this is related at all to inflammation. It'd be something that would be really interesting to look at.
What technology do you use to observe the ribbons?
Oh, okay. Yeah, I didn't really mention this. So we use two main technologies in this talk, or to generate the stuff in this talk. We used a confocal microscope, which is essentially at the light microscope level. And that is for any of the images you saw ribbons as tiny red dots. And then the images of ribbons that were at extremely high magnification, that was all done with an electron microscope.
Okay. And I think some of the other questions you kind of alluded to, just the path forward. Mm-hmm. Would you classify this as something related to neuropathy? Is there any relationship to that realm?
I'm not sure that I would call it neuropathy. I think that this is coming from another term that's often used in the field, which is called synaptopathy. So when people describe the loss of ribbons for inner hair cells, they often use the term synaptopathy. And so in my mind, neuropathy, anything with pathy on the end, would mean that you are losing something, losing the function of something, possibly. And in this case, I would say this might be closer to something like plasticity, where you're changing the characteristics of something. And we're not sure. And there is one other thing that I'll mention from the paper, which we also haven't looked into, but really interested in, which is there limited observations. It seems like there could be a increased branching of type 2s after noise damage. And so that would be kind of more in the realm of plasticity, again, something changing for the neurons.
Okay, great.
Mm-hmm.
A number of other questions and some under chat as well as Q&A. So Megan, if you have time, you can answer any of those or respond to the chat. Mm-hmm. We really, really appreciate you coming on as well as Arnold today. Two amazing